Us as we speak.
Fantastic. Well, good morning, everyone, and welcome to our second webinar. Our webinar series obviously is devoted to bringing key experts that we think are particularly relevant to what scleroderma patients should be tracking and should be interested in terms of emerging therapy opportunities. My name is Luke Evnen, and as some of you know, I was diagnosed with scleroderma in 1998, and shortly after joined the Scleroderma Research Foundation board. And over the past 11 years, I've been a board member, and beginning in 2002, I became chairman of the Scleroderma Research Foundation. The foundation is headquartered in San Francisco, and today we are America's leading nonprofit investor in scleroderma research. And in each of the past 5 or 6 years, and going back now for almost 25 years, we've been sponsors of research, and more recently in excess of $1 million a year. And I'm proud to state that the SRF has raised over $31 million in its history. We've consistently invested in excess of 80% of those monies on a year-in and year-out basis into our research. The very good news, and brings us straight to today, is that we are in a period of explosive growth in both basic and applied scleroderma research. There is newfound and profound interest from the pharmaceutical industry in scleroderma, and this is a dramatic change. Scleroderma is, of course, a rare disease, and until quite recently was almost totally neglected by industry. However, today there are more than 30 clinical trials taking place, and new and hopefully improved therapy is being trialed in pulmonary hypertension, where some fantastic advances have already been made. But beyond that, companies are also working to develop antifibrotic agents, immune system modulators, and other therapies. And today, we will focus on fibrosis with one of scleroderma's foremost experts. Just a brief word for some background. The Scleroderma Research Foundation webinars are scheduled to last 1 hour, and within that time we'll include a question and answer session following the presentation. Webinars are broadcast live and are also recorded for later viewing on the Foundation's website. Questions can be asked by using the chat functionality in the conference window. Please keep in mind that our conferences are for general information, and so we will not be able to address personalized questions and certainly not be able to provide personalized medical advice. It's wonderful to have all of you with us today, and it's my pleasure to introduce a gifted man who has really dedicated his career to helping scleroderma patients. Dr. John Varga is an internationally recognized expert in the diagnosis, treatment, and study of scleroderma. He is the director of the Northwestern Scleroderma Program in Chicago. Among numerous professional distinctions, he chairs the Scientific and Medical Advisory Board of the Scleroderma Foundation and is also a Scleroderma Research Foundation-funded research investigator. He was recognized as one of 50 heroes by the Arthritis Foundation. Dr. Varga is leading high-impact basic and clinical research, and his NIH-funded research laboratory was recently awarded, in collaboration with several other labs, an $11.3 million grant from the National Institutes of Health for a 5-year study on scleroderma biomarkers. A prolific researcher and author, he has published numerous research and review articles and 3 books on fibrosis and scleroderma. His most recent book, Scleroderma, From Pathogenesis to Comprehensive Management, was co-authored with Scleroderma Research Foundation-funded investigators, Drs. Christopher Denton and Fred Wigley. Dr. Varga's lab focuses on fibrosis and tissue repair. We welcome him today to discuss accelerating scleroderma research toward a cure. Thank you very much for being with us here today, Dr. Varga.
Good morning. Thank you very much. It's really a great pleasure to be speaking with you this morning, and as Luke has indicated, this is really a uniquely exciting time in scleroderma because of the confluence of a number of different things that I'll just briefly allude to. I think we're in a unique position to to really start thinking about breakthroughs in this very frustrating and difficult disease. Let me just see. Okay, so what I will try to do very quickly is give a little brief overview of where we are and what the current challenges have been in our ability to develop really breakthrough therapies for this disease, and what are the research opportunities and how I think applying modern technologies and modern know-how, we are really very well poised, as Luke mentioned, in the next certainly 5 to 10 years to develop really effective drugs that will hopefully really change the landscape for this disease. So let me just start by, seeing where we are. So if we think about the major complications of scleroderma, obviously it's a very diverse and multifaceted disease. But I listed here some of the most vexing and most serious complications that we deal with. And these are listed here. And where are we in terms of therapy for these complications? And you can see that we've actually, over the past few decades, have developed some very effective treatments for Raynaud's, and this is much less of a problem than it used to be. But for those patients who develop severe ulcers in the fingertips, we still are lacking truly effective therapies. We've made a huge progress in gastroesophageal reflux and heartburn and those complications where most patients now can be very appropriately and efficiently managed. The heart problems is an area that we're just becoming really much more aware of, and because the pathogenesis of heart problems is not really that well understood, I think we still have a long way to go in thinking about how to treat that. As Luke mentioned, a major advance has been made in terms of treatments for pulmonary hypertension. But a problem there is that many patients with scleroderma have not just pulmonary hypertension, but in combination with interstitial lung disease. And we're learning that the classic effective therapies for pulmonary hypertension may not work quite as well in scleroderma patients who also have lung disease, so this remains a significant challenge. In addition, renal crisis, which used to be really the most devastating complications of scleroderma, has significantly improved treatment with some of the newer drugs that have been introduced. But in fact, many patients still go on dialysis, so it still remains a fairly significant challenge to meet. And just to illustrate where we have been until recently, here is a slide that illustrates the outcome of a multi-center study focusing on treating lung disease. And in this study that was recently completed, patients were either given Cytoxan or placebo. Now, all of these patients had significant lung disease. And in this study, we monitored what happened to their lung function over time. And you can see that the patients who got Cytoxan, as I'm showing with this red dot, actually got better compared to the placebo patients who did not. But I think it's important to keep in mind that if you look at the difference between the two groups, it's actually not very significant, not very clinically meaningful. And furthermore, over time, as this treatment was stopped, even the treated patients went back to the untreated. So these results clearly indicate that Cytoxan, for example, is one therapeutic option, but it's certainly far from being a magic bullet that we need. And in fact, if you look at the outcomes of scleroderma until at least 2000, you can see that the mortality rates have, at least until that period of time, have not shown a great improvement. In this study, these investigators looked at a series of clinical studies, these are all clinical studies, over 4 decades, and looked at what's called the standardized mortality rate, that is, mortality of people with the disease compared to the control population. And in all of these studies, it significantly increased, and at least, again, until 2000, it hasn't really shown much change. Now, I think it's clear that in the last decade we've actually really begun to make some significant progress, and that may not be apparent for a number of years. But until recently, these challenges remained. And so if I were to think about where we are currently with treatment of scleroderma, I would have to say that we have developed, by and large, quite effective and safe drugs for many of the organ-based complications, such as reflux and Raynaud's. But we're still lacking truly safe and effective disease-modifying treatments. Now, I put in question mark there the HSCT, which stands for hematopoietic stem cell therapy, which is a bone marrow transplant. As you know, it's still an experimental procedure, but there are certainly case reports in small series suggesting that this could be highly effective, but I would say we still don't quite understand how it works. We don't fully know the mechanism of action, and we certainly don't know who are the appropriate patients. So at this point, it's fair to say that we clearly need to develop more effective and safe treatments for scleroderma. Now, what have been the obstacles? Why has scleroderma lagged behind many other diseases where the last decade or two has seen dramatic progress? And these things are very important, and they're becoming clear. We're increasingly recognizing that scleroderma is a very heterogeneous disease. No two people with scleroderma are truly alike. And this is very similar to the insights that our colleagues in oncology, for example, when they realize that cancer in the breast is actually many different types of disease, and lymphoma, and different patients with different types of lymphomas actually will need different kind of treatment. Another problem has been that we actually don't have good ways to measure whether the disease is getting better or worse. And listed here, you see a number of things that we routinely measure, and many of these do have some clinical validity, but overall they have not been well validated as truly reliable outcome measures. So these are challenges, but I would say they are not major challenges, and they are clearly being met with more research. Probably the most significant challenge is that we still don't truly understand the pathogenesis of this very complex disease. We don't generally know what causes it, and we're just beginning to understand some of the genetic basis. But these areas are seeing a great deal of progress. Now, let me just skip ahead here. This slide kind of gives a timeline of how research in scleroderma has evolved over the past really 100 years. In the beginning, there was the classic observational research where clinicians and pathologists would look at the scleroderma and describe what they saw, what we call empiric research. Over the next couple of decades, and increasingly the 1960s to 1980s, research, what's called reductionist approach, which is hypothesis-driven, has emerged and really has started to really shed light on some of the mechanisms and some of the underlying pathways that cause scleroderma. And then increasingly the development of animal models, although we still don't have a true animal model of scleroderma, but research in mice and other animals does now start to allow us to look at certain specific pathways and molecules. More recently, and I think the most promising, is this area down here of the application of translational research, that is the bench-to-bedside research, and the application of what one might call unbiased or high-throughput discovery research, where the investigator doesn't have any notion of what the question is, but rather looks at everything that we can measure in people with scleroderma. And this includes genetics and RNA and proteins, and putting all that information together in a large algorithm that could be described as a systems biology approach to try to understand from this type of approach what are the critical pathways. And so there's a veritable tidal wave, particularly in the last 5 years, of information that's coming out of all these research activities. And in fact, the challenge and opportunity right now is to really understand what is all this data telling us, what's important, what isn't, and ultimately how do we manipulate and understand the data to be able to transform it to clinically meaningful change and understanding. So there's been a tremendous progress in scleroderma research, and the causes, some of them are listed here. There's an increasing awareness of this disease, which is a very good thing, and some of these international research workshops, the World Congress of Scleroderma, the annual research workshop funded by and supported by the Scleroderma Research Foundation have been very, very instrumental in both increasing awareness and bringing people together. As Luke mentioned, there's also now really, for the first time, interest from the pharmaceutical industry. They used to have very low interest rate in rare and orphan diseases, but that's clearly beginning to change. There's increasing connectivity among researchers where, particularly because of the internet, people from all around the world can actually work together in integrated research teams. And this is also impacting on young investigators that are critical to maintain the momentum in research, and we see more and more young physicians and scientists pick scleroderma as an area of great interest to study. The application of these high-throughput technologies like microarrays that I'll mention and large-scale genetic studies where you can study thousands and hundreds of thousands of genes in very large groups of patients really is giving us a unique new insight into this disease that was just not technologically feasible before. And all of this is actually leading to a very nice scientific integration where investigators from various fields like cell biology and molecular biology, genetics and genomics begin to talk to each other and work together to develop a systems biology, which is really a holistic or more integrated way of looking at the disease. So with that background, let me start focusing a little bit specifically on scleroderma and scleroderma research. Now, I think at a very simplistic basis, we could envision that in a genetically susceptible individual, a person who has the appropriate genetic risk factors, particular kind of injury, which could be, for example, a viral infection, but it's generally not well known, leads to a number of responses. And these in scleroderma include autoimmunity with the generation of autoantibodies, and this is just a picture of what autoantibodies look like under the microscope, a vascular disease with Raynaud's and digital ulcers, and ultimately this kind of obliteration and closing off of the small blood vessels that you see here in this pulmonary artery, for example. And over time, these processes lead to what is probably the most striking and most intractable complication of scleroderma, which is fibrosis, also called scarring. And you can see in this scleroderma skin biopsy, the normal skin is really pretty much replaced by this dense collagen and connective tissue. And this is really what accounts for the hardness and thickness of the skin. And this fibrotic process is actually very similar to what can happen in people in the lungs as well. So fibrosis is really the key challenge and the key target complication that we need to think about in treating scleroderma. And so the key questions that many people have asked are, what causes fibrosis? How does it develop? What are the genetic risk factors? And ultimately, can fibrosis be prevented, halted, or even reversed? So let me just give you a little bit of a history on this. And this work started in the 1980s, and the approach that we and many investigators around the world took is to take biopsies from patients with scleroderma, take skin biopsies, grow the cells from the skin in these petri dishes as shown here, and then dump in a variety of different proteins and growth factors, and essentially determine what happens when these cells normal or scleroderma fibroblasts are stimulated in the Petri dish, what happens to their collagen production, which is really what ultimately causes the fibrosis. And the finding that we made in the mid-1980s with Dr. Sergio Jimenez, who was my mentor and is one of the leading scleroderma researchers in Philadelphia, is that there was a substance in pig platelets, and initially we had to use a very large number of platelets to get this purified growth factor, that acted as a very strong stimulus to stimulate the fibroblasts. And when these fibroblasts were exposed to the growth factor, they made huge amounts of collagen. In other words, they behaved like scleroderma fibroblasts. Now, when you examine the proteins that these fibroblasts make, you run what's called a gel, a protein gel, and that's what this looks like. And this is proteins made by healthy normal fibroblasts. And this is proteins made when these same fibroblasts are stimulated by TGF-beta. And you can see that many of these bands, particularly the collagen bands, are very much darker. And this really indicates that this growth factor actually stimulates the fibroblasts to become scleroderma fibroblasts. So the growth factor is TGF-beta, and over the years it's become very extensively studied. And what these studies showed is that TGF-beta is a critical molecule to regulate tissue repair. Now, it's very important in normal wound healing. So here, if you have an injury to your skin, there is rapidly repair of the normal skin structure. And this is driven by the cells that are fibroblasts and is stimulated by TGF-beta. So TGF-beta is very important in normal wound healing. However, if TGF-beta is abnormally regulated and essentially there's too much TGF-beta being produced, then instead of a normal tissue repair to some types of injury, you get this very exuberant and abnormal, exaggerated kind of repair. You can see that there's a huge number of fibroblasts, and more importantly, this substance, which is really the collagen that builds up, is massively overproduced and deposited in this tissue, and this is really what leads to fibrosis. So whether one looks at the skin or the lung, you see the same excessive collagen accumulation in the tissue. So TGF-beta is very important in driving this, and in fact, it does appear to be important in scleroderma. In this picture, I'm showing skin biopsy from people with scleroderma. Now, normal skin should have no TGF-beta at all because it's not active unless you have an injury. But what we found is that in people with scleroderma, oftentimes in the skin biopsy, we see these cells such as shown here, or a different method using to detect TGF-beta here. There are fibroblasts that are showing high levels of TGF-beta. Now, TGF-beta can actually directly cause fibrosis, and if one makes a mouse that overproduces TGF-beta, what you find is that compared to normal skin, where there's this thin layer of collagen indicated by the blue, the mice that make too much TGF-beta actually develop what really looks like scleroderma. So there's this huge accumulation of collagen replacing the normal structure of the skin. So TGF-beta seems to be capable of causing what looks like scleroderma both in fibroblasts in the Petri dish as well as in mice. So our research and many other laboratories then over the following decade really focused on trying to understand how does TGF-beta work. So if these dots are the TGF-beta molecules, we recognize that they must bind to a receptor on the cell, as shown by this structure, and that receptor then must then send signals into the cell that lead to increased collagen production and fibrosis. But for many years, this really remained a black box. Over the past decade or so, however, there's been tremendous progress, and a more sophisticated picture of understanding how TGF-beta works is shown here, where we now know that TGF-beta binds to the receptor. It activates a series of molecules as shown by these different color ovals, and we've identified many of the molecules that are activated by TGF-beta. These include, importantly, a molecule called c-Able that I'll talk about a little bit, and other molecules such as SMADs, EGR1, and P300, and so on. Now, these are very important to understand because knowing their identity allows us to, number one, measure them, and number two, manipulate them, whether it's using drugs that inhibit their function or alternately making mice models where we inhibit or overexpress these molecules. So having this information really gives us good tools to start understanding the function and manipulate the function of TGF-beta in scleroderma. Now, the question has come up, though, more recently, is TGF-beta involved, and is it actually important in scleroderma? And perhaps more importantly, is it important in all the patients with scleroderma, or is it that it's important in some people who have the disease but not in others? A very important question to ask, because that will allow us to potentially think about which patients need to be targeted with anti-TGF-beta therapies. And the approach that Mike Whitfield, who is one of the leading investigators funded by the Scleroderma Research Foundation, has taken is to essentially study all the genes in the scleroderma skin and identify unique patterns to be able to determine which patients are really showing the TGF-beta response. And let me just jump ahead and show some of Dr. Whitfield's results. What he's been able to demonstrate is that about half the patients with scleroderma in this particular subset of skin biopsies, where you see a lot of red, it means that the TGF-beta pathway is really turned on. So you can see that these patients have a very strong what's called TGF-beta signature and potentially could respond to therapies that inhibit TGF-beta function. Now, in contrast, here is another group of TGF-beta patients, and they are mostly green. And that means their TGF-beta is actually not activated. And one could speculate that these patients probably would not benefit from treatments that target TGF-beta. So this is a very important insight that still remains to be tested in the clinical setting, but I think is a major advance on how we think about individualizing therapy and thinking about personal medicine. So TGF-beta is clearly important, and this slide just indicates that we now have compounds and chemicals and potential drugs that can inhibit TGF-beta signaling at a variety of different levels. I think all of these will ultimately need to be tested and evaluated to see which one is most effective and which one is safest. Now, I just want to very quickly mention an important new drug discovery paradigm, and that is what's commonly called drug repurposing, because this has important implications for research. And the problem here is that it's become apparent that to develop new drugs is phenomenally expensive and takes an incredibly long time. So it is now typically up to 15 years and can cost up to $2 billion to bring a drug from the initial discovery right into the clinic. And at the same time, despite the increasing cost, unfortunately, the number of drugs that have been FDA approved during the past 2 decades has actually been stable and even perhaps declining. So, and this is because a great majority of drugs do not reach the clinic. So we need an alternate approach, and that approach is what's called drug repurposing. And that essentially means to take drugs that are already approved for other clinical indications and introduce them to appropriate clinical settings. This, for example, shows a number of drugs that are currently in use for various indications such as leukemia and diabetes that, based on the research, might work in blocking TGF-beta and might be effective in treating a disease like scleroderma. Now, let me just give you very quickly a summary of the story of Gleevec, because I think this is a nice illustration of the drug repurposing strategy. So Gleevec is a drug that was discovered to target a abnormally expressed molecule called c-Abl. C-Abl is a kinase, and this kinase, when it's upregulated and mutated, can cause leukemia. The drug c-Abl or c-Abl inhibitor or imatinib was specifically developed to block this enzyme, and it was so dramatically successful in the treatment of leukemia that it made the cover of Time magazine in 2005 as a magic bullet in a highly specific treatment of a very deadly disease. And in fact, what happens with c-Abl is that we have a signaling pathway that leads to the development of leukemia, and if we block this signaling pathway with Gleevec, the leukemia is essentially completely arrested. Now, what does this have to do with scleroderma? So a seminal publication came out a number of years ago showing that the same drug, Gleevec, can inhibit the fibrogenic activity of TGF-beta, which is exactly what we'd want, and can prevent the development of scleroderma-like changes in the mouse. In other words, what this investigation and subsequently many others confirmed showed that the same pathway that seems to cause leukemia under some conditions can lead to fibrosis and scleroderma under other conditions, and that presumably blocking that pathway using Gleevec might also be effective in treating fibrosis. And in fact, these turn out to be the case. These are just models in the mouse looking at lung fibrosis and skin fibrosis, very much like what you see in scleroderma. And what this shows is that in the mice that received the Gleevec, as shown in these red brackets, the lung fibrosis and the skin fibrosis were markedly attenuated. So based on these findings, we and others have organized a small clinical trial. This was an open-label study of patients with early disease. Open label meaning that there was no control, no placebo control. Everybody got the drug. And a total of 27 patients were enrolled, all with early disease. And one of the things that you can see is that this drug is not terribly well tolerated in a lot of people with scleroderma. Almost half the patients had to drop out due to various side effects or other problems. What happened when the drug was given, though? And this is the outcome of the results of the skin score, one way of measuring skin fibrosis. And essentially, we see a modest decline over 48 weeks. Now, this is not a huge improvement. It would be nice to see a dramatic decline, but at least it shows that at the very least the drug prevented the disease from getting worse and in fact potentially led to a modest improvement. Of course, we'd like to see a greater improvement. What happened in the lungs? The same story. Very little change over 48 weeks in the different ways of measuring lung function. And you could say, well, this is a little bit disappointing, but on the other hand, one could argue that these patients without treatment may have shown a deterioration over time. And so Gleevec appears to be, at the very least, capable of stabilizing lung function and preventing its progression. So the story with Gleevec right now is that we have a drug that's already FDA approved, and we know a lot about this drug, and that small clinical trials in scleroderma show that it's at least able to stabilize the disease. Now, these are not perfect studies. They are small, they are of short duration, and they're open label, so I think clearly more work will need to be done. Let me just end by mentioning another very interesting drug repurposing strategy very quickly, because I think this is another drug that you'll hear about. Bortezomib or Velcade is a proteasomal inhibitor. It's a highly engineered and very specific drug that was approved in 2003 for the treatment of multiple myeloma, a very bad kind of leukemia where this drug was shown to be quite effective. And over the following years, information showed that other forms of malignancies can also be effectively treated by this drug. So we did some animal studies looking at the efficacy, again, using this mouse model of scleroderma. The mouse are given bleomycin, and then they are treated with the drug to see what happens. And what the results showed is that administering bortezomib to the mice pretty much prevented the development of the severe scleroderma-like changes in the skin, so it's markedly attenuated. And the results were even more promising when we looked at the lungs. So mice given bleomycin developed severe scleroderma-like lung fibrosis, as you can see here with the blue color, whereas the mice that got bortezomib, the lung fibrosis was almost completely prevented. So these are very promising preliminary results, and we are actually now in discussion both with the federal government and drug companies to design the first clinical trial of bortezomib in scleroderma. This will be a randomized controlled study, and these studies initially are usually relatively small because we want to focus on understanding the efficacy, but also very importantly the safety of this drug, because we don't know yet how people with scleroderma will react to this drug that's really being used for multiple myeloma. So to bring this to the end, as Luke mentioned, this is a tremendous and unique time in scleroderma research, both basic research as well as clinical, and the number of new compounds, both brand new compounds as well as drugs that have already been used for other diseases being studied for scleroderma, is really exploding. And this slide just lists a partial list of drugs and interventions that either are already in clinical trials, that's indicated by these red asterisks, or are currently being planned for clinical trials. And I think we're going to definitely see very promising results and significant improvement with some of these agents within the coming 5 years or so. So with that, I want to thank you, and I think now we go back to Luke and take any questions or comments.
Well, John, that was really terrific and a really great overview. Much appreciated. So just from a process standpoint, this is the time when we would really welcome the chance to take some questions from you in the audience. We still have about 15 minutes, and we'll answer as many questions as possible. If you do have a question, please submit it by typing and sending it to us in the chat box, which you can find at the left of the main slideshow window. But while you're all composing your questions, why don't I just get started with going back to maybe one of your slides, John, about some of the stuff that's going on with scleroderma clinical trials, and ask you about a couple of the agents, and maybe you can help our audience understand where some other of the agents are being trialed, but to talk a little bit about the direct anti-TGF-beta monoclonal antibody, the blocking strategy, and also maybe if you can talk to us just a little bit about maybe the anti-CTGF, which are seemingly very direct ways to block TGF, and maybe you can initiate the conversation about where we might be headed with some of those strategies.
Absolutely. TGF-beta is this growth factor, and it has really become very clear over the past 2 decades that it's probably the fundamental driver of all kinds of fibrosis. So it's important not only in scleroderma, but lung fibrosis and kidney fibrosis, and even in cancer. So there are a number of companies and a number of strategies to block TGF-beta, and one of these promising strategies is an antibody that is being made by Genzyme, a company, a biotech in Boston. And they have, as I understand it now, some very good safety data, and are currently doing, I believe it's a phase 1 or phase 2 trial in a small group of patients with scleroderma, initially looking really at safety as well as biomarkers. So in these initial studies, you don't necessarily look at dramatic effects, because they might take a longer time and might take much larger groups of patients to look at. But really look at, is the drug well tolerated, and is it changing things that we can measure that would give us encouragement that the drug is really working and is likely to do what we think it might do. Another strategy to target TGF-beta, again, another biotech company, a different biotech in Boston, is to block the activation of TGF-beta using, again, an antibody. So both of these are given intravenously, and both of these are in very early stage. There is a study as well, basically a very similar thought on blocking another growth factor called CTGF, or connective tissue growth factor.
Great. We have one question from one of our viewers, listeners, is, John, can you address the results from basically the high-dose immunotherapy followed by stem cell transplant trials and the impact on fibrosis that those trials have shown, or and what the overall results are in your sort of current thoughts about that as a therapeutic option?
Sure. The idea behind, essentially this is stem cell therapy, or previously called bone marrow transplantation, but the basic idea is that some profound immunological abnormality may be very important in triggering the disease. And although immunosuppressive drugs like steroids typically don't work, the idea here is to virtually wipe out or paralyze the immune system and then push the reset button. So patients get very high-dose chemotherapy, damages or virtually wipes out their immune system, and they are given back, in most protocols, their own immune cells that have been harvested previously. There are other protocols of allogeneic transplant where matched donors are given, but the most commonly used type is the autologous, where you essentially get your own stem cells back. Now, it's highly experimental. There are 3 studies going. One recently published some very, very encouraging preliminary results in a small study. And in fact, in this study that was done at Northwestern, all 10 patients who got the bone marrow transplant showed a very good response. So this is encouraging. I think what must be thought and kept in mind, though, that this is a very, very, very aggressive and, of course, expensive therapy that can be associated with significant side effects and even mortality in some cases. So I think we need to be very careful about who gets this, how it's done, and what the appropriate follow-up therapy will need to be. Now, there are 2 large multi-center trials that are underway, and I think those results should become available starting next year. So I think that will be very important to look at whether this very encouraging small experience is really borne out in international multi-center studies.
Right, but my impression, John, is that the protocol that was used at Northwestern was an evolution of the even more aggressive protocol that was used in some of the international studies, so that I think some of the learnings about trying to reduce the toxicity of the actual induction part of the protocol, where they give the chemotherapy, they're trying to lessen effectively the immunosuppression so that the disease burden and the mortality risk is lowered.
That's correct. So, I think it's a lot like the evolution in cancer therapy, where many initial cancer treatments had horrendous side effects and even mortality. And as people begin to understand the right patient population and the right doses, the toxicities started to get better. And I think we definitely are seeing that with this type of approach where, for example, being very cautious about using radiation and some of these very toxic drugs at more carefully balanced doses seems to be leading to much less complications over time.
Great. So, John, let me take another question from the audience, and there's a question about how you basically monitor disease, and maybe we can put it in both a general and in a fibrotic context. Are there good blood tests? What are the ways to try and figure out, as we've talked about, the disease seems to have an uneven progression. So, can you speak to that issue both overall and with respect to, as an example, lung fibrosis?
Absolutely. This is you're absolutely right. I mean, this is one of the real difficulties. What do we use to measure outcomes? And currently, there's a set of tests and evaluations that are widely used, and certainly have some utility. Particularly, pulmonary function studies are quite reliable, and if they show an improvement in pulmonary function, that very likely does indicate stabilization or improvement of lung involvement. High-resolution CT scanning can be useful. Echocardiography is certainly very useful in those patients who are at risk for pulmonary hypertension. More research-wise, there's a lot of interest in looking at skin biopsies, for example, and looking at all the different genes that are being up and down regulated and how they change with treatment. And ultimately, one could do the same thing perhaps even in the blood so that it would be non-invasive. One other area that is getting a lot of attention is the most low-tech and simple approach, which is patient-reported outcomes. And by that I mean designing questionnaires that the patient can fill out, and when these are validated, can actually give us a very good idea of is a drug actually working and improving the disease or not. So there are some clinical tools that are quite useful, and there's a lot of research in this area to identify metrics and biomarkers that could be really highly useful to predict good responses.
Right. John, you briefly talked a bit about looking at gene expression levels. I think maybe just to sort of discuss the difference between scleroderma as a genetic disease from a predisposition standpoint, and then changes in gene expression as the disease basically comes on in your adulthood, I think might be helpful as a clarifying point for our audience.
Sure. Scleroderma is not a genetic disease in the sense of heritability. It is very, very unusual, perhaps about 1 in 100 cases where it actually is inherited. However, there are genes that are susceptibility genes, and this is true for many complex multigenetic diseases, so that can contribute to an increased risk. And it's well known, for example, that certain autoimmune conditions do tend to run in families, so Raynaud's and thyroid problems and so on. But scleroderma is not truly a genetic disease in the sense of the way we think about typical inherited conditions. When we talk about gene expression, what we're really talking about is looking at the affected tissue in people with scleroderma, and by analyzing the thousands of genes that are being up and down regulated normally and comparing them to controls gives us, number one, some insight into what are the different pathways and molecular mechanisms that cause the disease. And number two, they give a unique fingerprint of the molecular changes that are happening over time, so that they can be used, for example, to identify which patients might require drug A versus drug B, and also perhaps to identify whether a drug is working at the cellular level. And we might potentially see those changes at the cellular level even before a clinically meaningful response occurs. It might precede a clinical improvement.
Right. Okay. Are there any other questions from the audience? Please use your chat box. John, maybe the one thing, talk a little bit more about microarrays and disease heterogeneity. You did touch on it, but we have a couple more minutes, and I think it might be useful. Do you foresee an opportunity to try and group patients into sort of more homogeneous subcategories, if you will, and then run, if you will, clinical trials against basically these subgroups of patients? Do you envision that as a direction for future therapy or future clinical trials?
I think that's probably going to be the case. And again, I think if we think of leukemia or lymphoma, 20, 30 years ago, we thought they were just the same disease. And as the molecular techniques got more sophisticated, it became clear that there are many, many, many different types of lymphoma, different types of breast cancer, and patients with falling into the different subsets actually require very dramatically different treatments. Some people with breast cancer have a very, very benign course and good prognosis and require very mild therapy. Others probably need very aggressive therapy from the get-go because we learned from their molecular signature that these patients don't do well unless they're aggressively treated. And I think it's starting to emerge that it's the same scenario in scleroderma. And we know from clinical experience that some patients with scleroderma have a very benign prognosis, have a completely normal life expectancy, and really do very well with minimal complications, whereas other patients have a very aggressive and progressive disease. We know this from clinical experience, but we haven't really had sensitive and reliable predictive tools that would allow us to define these patterns from the get-go. I think with the application of technologies, and particularly microarray could be one of those, we might be in a position within the next couple of years where we can look at a patient who just presents with first onset of scleroderma and really with a few tests be able to very accurately predict whether they have the severe progressive type or the different subtype, and therefore target and tailor therapy individually to that patient with a high degree of confidence that we're not under-treating and not over-treating, but giving really the right type of personalized therapy that's appropriate for his or her type of scleroderma.
And John, do you envision that's a blood test, or will that be a tissue test? I mean, what's your best guess of what format that test might take?
Well, currently the people who are the leading investigators in this field, such as Mike Whitfield, feel that it's tissue-based, but I think hopefully, ultimately, as the technologies develop, we will be blood-based because you always prefer non-invasive tests to tests that require a biopsy.
John, one thing we sort of, just to go back to TGF-beta expression and the signature that you showed, has there been work done to correlate disease progression in the lungs with that TGF-beta signature? Or to state it a different way, do we know whether or not patients who end up with a scleroderma lung fibrosis are the same patients who have basically the upregulation of the TGF-beta signature?
I think the finding of the TGF-beta signature, that slide I showed, is in a way the poster child for the potential utility of this approach, because in fact that's exactly what we found in this project, that the patients who had the TGF-beta signature significantly were more sick and had a more aggressive disease and more lung disease than the patients who did not have the TGF-beta signature. And so this is a very preliminary but a very powerful illustration that just by looking at gene expression signature, we might be able, in fact, to predict not just what happens in the skin, but even in internal organs. And really would lead you to think that patients who have this signature may be the patients where we might use early aggressive therapy targeting TGF-beta, so that this approach might really lead to a very sophisticated, personalized medicine treatment approach.
That's great. Well, John, I think we've actually consumed basically our allotted time. I really want to thank everyone today for joining us and for participating in our second webinar. We very much appreciate some feedback. So when you do close the window, there will be an opportunity for you to provide some comments and some opinions for us. In particular, I'd really like to thank Dr. John Varga for his commitment to us today and for his decades-long service to the scleroderma patient community and research community. So, John, thank you very much.
Thank you.
As a reminder, the entire webinar will be available for a download beginning next week on the Scleroderma Research Foundation website. For more information or to make a donation to the Scleroderma Research Foundation, please visit us at our website. That's www.sclerodermaresearch.org, or shorthand www.srfcure.org. Our next webinar is scheduled for 9 a.m. Pacific Time on Friday, February 10th, and you'll receive an email in the few days to come to register for this, the third in our series. Again, thank you all very much for dialing in. We look forward to seeing you again soon.
Thank you. Please stand by.